welcome to my channel, and today's topic is excitation-contraction coupling theory. So before we start with this topic, let us revise some of the previous topic which is going to help us in understanding the excitation-contraction coupling theory. So let us revise the sarcotubular system. So as you can see in this diagram that I have shown one of the myofibril. So let me mention it, that this structure is myofibril, and this is the sarcotubular system. Actually, for understanding purpose, I have drawn this diagram like this. Otherwise, this whole sarcotubular system is over this particular area. So, this is the sarcotubular system. The outermost layer, it is termed as an sarcolemma, which is a plasma membrane. Sarcolemma, which is the outermost layer. Now, in this, as you can see, these structures. So, these transverse tubules are known as an T-tubules. These transverse tubules are known as an T-tubules, and the longitudinal one is known as an L-tubules. I have explained this thing in my previous video. Now, for the revision purpose, let me explain this again, that these are the T-tubules as they are transverse and they are penetrating inside the muscle. So, one end of the tubule, as you can see in this diagram, one end of the tubule is opening in the sarcolemma and the another end inside this myofibril. So, it is penetrating through and through. Another thing is L-tubules. L-tubules, as they are longitudinal in nature, they are called as L-tubules, and they have bulge portion on both of its ends. So this bulge portion is known as an cisternae. Bulge area is known as an cisternae of the L-tubules. Now, as you can see that there are two cisternae, and in between there is presence of T-tubule. So these three things. So number one is cisternae, then comes the T-tubule, and then again cisternae. So two cisternae and in between T-tubule, this is known as a T-triad. Now, let us focus on the functions of this T-tubules and the L-tubules. T-tubules transversely placed inside the myofibril, and as the one opening is over the sarcolemma of the T-tubules, so whenever any extracellular fluid is present or an action potential comes, so it will be directly reached to the inside of the myofibril through the T-tubules. So T-tubules are actually making the work of the extracellular fluid as well as the action potential very easy, that it can reach to the depth of the myofibril very easily. Now comes the L-tubules. L-tubules are the longitudinal tubules, and this cisternae contains a large quantity of calcium ions. So the calcium ions, when the action potential reaches up to this particular level, so those calcium ions are released into the sarcoplasm and they initiate the contraction mechanism. Now we come to our topic, that is excitation-contraction coupling theory. So as we know that the calciums are present in the cisternae and they are released when the action potential comes. But before that, we must understand the molecular, or I can say the contractile elements of this myofibril. So as I have discussed in my previous video, that electron microscopic picture of a myofibril shows there are A band, I band, Z line, sarcomere, that all we have discussed. But actually the depth of this concept is the contractile elements of the myofibril. So as you can see in this diagram, so these two are the contractile elements. So A band, so as you can see in this diagram, the black line, these are the A bands. A bands, they have a protein named myosin, and the I band, that is the red lines. So I band, they have three types of proteins. First is the actin, second is tropomyosin, and the third is troponin. Troponin has three subtypes, that is troponin C, which binds with the calcium, troponin T, which binds with the tropomyosin, and the troponin I, which binds with the F-actin. Now what is F-actin? F-actin is basically the fundamental portion of the actin filament, and which is actually the polymer of G-actin. Now let me explain you this thing, that when multiple G-actin monomer, they are combined and they form one polymer in the form of a filament, is known as an F-actin, or we call it as an actin filament. And this actin filament has an active site to bind with the myosin. Similarly, myosin has two active sites. First is to bind with the F-actin, and the second is for the ATP. So let us understand this myosin molecule. So as you can see in this diagram, that this is the myosin molecule, and in this myosin molecule we can identify two structures. First is this one, that is the globular head portion, that this much area is the head portion, and a long tail, which is double helix in nature. So two portions of the myosin filament, that is head portion and the tail portion. Now comes the actin filament. In the actin filament, as I have mentioned, there are three different proteins. So the circular one, as you can see that I have explained that F-actin, that is, it is a polymer. So the circular one, that is the F-actin. A red line which you can appreciate over here, that is the tropomyosin, and this structure, this typical structure is the troponin with its three subtypes, that is troponin C, troponin T, and the troponin I. So these are the contractile elements and the proteins which are there in the electron microscopic picture of the myofibril. I hope this concept of sarcotubular system as well as the contractile elements are clear to you. Now let us understand what happens when an action potential arrives to the sarcolemma. Now let me rub all these things. So I'm going to draw a small diagram which will be very much helpful for us to understand the excitation-contraction coupling. So for that, this is the A band. A band has a myosin protein, and the myosin has portion, that is the head of the myosin. So this is the head of the myosin. On the other side, let me draw I band. Again the A band. Now I'm going to draw a little bit in detail that this is the perfect head of the myosin. And again the I band. So let me draw it like the real one. F-actin, then comes the tropomyosin, and then comes a large portion of troponin. This is the F-actin, tropomyosin, and a large portion of troponin with its three subtypes. So now, as we have understood the sarcotubular system, so suppose this is a T-tubule. On both of its side we have cisternae, that this is the cisternae. So when an action potential arrives, and reaches up to the T-tubules, from both the cisternae there is release of large quantity of calcium ions. So this is a T-tubule, this is L-Tubules, this is the cisternae. From the cisternae, there is the release of calcium ions when an action potential arrives. This calcium ions, they are going to bind with the troponin C. Calcium ions, they are going to bind with the troponin C, which is present in the actin filament. Now, in the normal condition, what happens is that tropomyosin, this line, tropomyosin is actually covering the active site present over the F-actin. Remember, I have just explained that F-actin has an active site which is going to bind with the head of the myosin. But in normal condition, it is covered by another protein, that is tropomyosin. Now, when the calcium ion are released from the cisternae, they are going to bind with the troponin C, and a structural changes are taking place, and they are going to change the shape of the actin filament, and the active site for the F-actin will be uncovered from the tropomyosin. So now we have exposed F-actin site. Similarly, this is going to happen it over here also. And myosin head is already ready to bind with the active site of the F-actin. But this process is actually a stage wise. So let me explain you what is happening. So what happens is, this is the polymer of F-actin and this is the myosin head. So myosin head, they first bind with the active site of the F-actin. Remember, myosin has also an active site to bind with the F-actin. F-actin has a site to bind with the myosin head. Now they are attached to each other. Second thing what happens is, this myosin head, it has a portion that we call it as a head, then comes a hinge and the arm. So hinge means it can turn. So what is happening when they are bind with the F-actin? So it is going to turn like this. So as you can see the change in the position, that the head portion is directed towards this direction, but when it is attached with the F-actin, it is going to change its direction, and this direction is towards the Z zone, or I can say the H zone, which is there in the middle of the A band. And the change in the direction of the head, or I can say the tilting of the head, is known as a power stroke. Is known as an power stroke. So this is very, very important for the contraction, as this power stroke, which means that this myosin head is attached with the active site of the F-actin, and it is going to tilt towards the midline of the sarcomere, and this tilting is actually pulling the F-actin towards the midline, right? So this is the myosin head, and if it is tilting towards this direction, so ultimately the actin filaments are going to pulled towards the centermost H zone. And when this is going to happen, a complex which is formed, which is known as an ectomyosin complex, that actin filaments, they are completely attached with the myosin heads. This is known as an ectomyosin complex. Let me write it over here, the important point, that is ectomyosin complex. And when the maximum contraction inside the muscle is achieved, it is said to be due to the ectomyosin complex. And histologically, the ectomyosin complex is the first sign by which we can tell that the muscle is in a complete state of contraction. So again, let us revise the whole procedure. That action potential arrives in the T-tubules, and from the cisternae, the calcium ions are released. Calcium ions are going to bind with the troponin C. This troponin C, there is some structural changes are going to take place, so that the active site of the F-actin will be uncovered, and they are already ready to bind with the head of the myosin. When they are attached with the head of the myosin, so head of the myosin has three portions, that is head, hinge and the arm, and it is going to change its direction, right, from this direction to the direction which is there in the exactly towards H zone, or I can say the centermost portion of the sarcomere. And this procedure also requires energy. Now energy comes from the ATP. So ATP molecule which is used in this, there is one side of the ATP, so that ATP molecule is used and it is catalyzed into the ADP and the inorganic phosphate with the help of an enzyme, that is ATPase. So this also requires energy, and ATP is catalyzed into these two portions. Now comes the most important thing, that is we have understood the contraction of the muscle, right? But the relaxation is equally important, because relaxation, if relaxation doesn't occur, muscle will remain in its shortened position, right? So for the relaxation, what happens is the calcium ions which are already attached with the troponin C, they are going to detach from that troponin. And as they are detaching from the troponin, again structural changes are going to take place, and the tropomyosin, this red line, tropomyosin, is going to cover the active site of the F-actin. So as the active site over the F-actin is now covered, myosin head cannot bind with the F-actin, and there is going to relaxation of the muscle, or I can say the muscle is going to assume a position which I can call it as a pre-contraction state. So that is, the pre-contraction state is achieved, and this theory is known as an excitation-contraction coupling theory, or you can say a ratchet theory or a walk-along theory, in which how does a contraction occurs as well as the relaxation of the muscle is very well explained. Now again revise both of these things. First is excitation. Action potential arrives at the sarcolemma at the level of T-tubules, going to contract the muscle. For the relaxation, calcium ions are going to detach from the troponin, and those calcium ions, they are again pumped back into the cisternae of the L-tubule. So calcium ion from here, they are going back towards the cisternae, and there is less calcium ion concentration in the sarcoplasm, and this is going for the relaxation state. So this was all concept regarding the muscle contraction. I hope you like my content. If you like, do like, share and subscribe my channel. Don't forget to hit that bell icon and share these videos maximum, so that one can understand the topic in a very simplified way. Thank you so much. Thanks for watching the video till end.